All right, then let's go ahead and start things off. Today is Tuesday, April 27th, 2021. We are playing some catch up today. We need to finish off the rest of our brain anatomy, which is where we'll start. We'll start in lab. Then once we get that done, we'll talk about spinal cord. And while we talk about the spinal cord, we also still have to talk about the protections of the brain and spinal cord. So we definitely need to talk about those things as well. And then we will make sure we have time at the end to talk about our cranial nerves. Remember, as we've talked about, those cranial nerves, especially where we are right now, could be as much as about 25% of your lab exam. And of course, it should go without saying, anything that is that important on your lab, could there find some questions, can some questions on it find their way onto your lecture exam score, into your lecture exam? Yes, absolutely. You have 3 more assignments that you're going to be turning in, only 3 more chances to get those free points. The first is your 15-point nervous review that, again, will be graded for correctness. Hopefully you have slowly started working on those. If you haven't, that's okay. The stuff we talk about today will okay. Will definitely help you to complete them. So again, because we're talking about, it's really about that relationship between structure, function, and location. So we will definitely get to that stuff today to help you with that. And again, one of the things you may notice as you go through it, there is some redundancy on the answers to those questions. And that's the point, is because I want you to really emphasize that when you know the function of something, you know the structure of something, you know the location of something. And one of those pieces of information can help you with the other 2. So that is the point of that review. Then you have 1 more unit review 14, and 1 more home lab that you'll be doing. I want you, it's a fun lab, the Reflexes Lab. There's a couple activities in your lab manual that I want you working on. Looking at the cranial nerve and spinal nerve reflexes. I want you to complete all the activities. Now, some of the activities may involve things like a pen light or a flashlight, which hopefully most of you have. But others could involve things like having a tuning fork. If you don't have a tuning fork, that is okay. But what I want you to do is complete the activity as a thought experiment, or look up online on the almighty Google and figure out what's going to happen. And I want you to write down what you expect to happen, or what you think to happen, or what you looked online and found out what is going to happen. I do not want anyone writing, I could not do this activity because I did not have a tuning fork. Right? That's not acceptable. You will lose points for that. I want answers. So if you can do the activities, and many of them you will be able to do, that's great. It's fun. Do it with your loved ones and other people in the house. But if there's any that you can't do, then like I said, answer the questions as a thought experiment, or figure it out online what you think would happen and write that down that way. So do as much of it as you can, and then the rest I want you to either, again, you're not going to get wrong for guessing wrong. So you don't have to look it up on the interwebs. You are more than welcome to just write down what you think would happen. Just make sure you justify your explanation as what you think would happen. And that's all I'm looking for. I'm looking for you doing it as a thought experiment. If you want to have the right answer and want to look it up on the almighty Google, you are welcome to do that. And that is due on Thursday the 6th. Again, there are basically 2 activities. Procedure 2 on pages 371 through 374, and procedure 2 on pages 380. So it's from a couple different units, because it's both cranial nerve and spinal nerve reflexes that you'll be doing. And then, as we talked about, all labs all the time. I mean, all exams all the time. Tuesday the 11th, your 5th lab and lecture exam, just like we've talked about before. And then, of course, your final exam, which, again, is going to be 100 multiple choice questions, cumulative, everything we've covered on the class. It does count as a grade on its own, but remember also it can replace your lowest lecture score, not your lab scores. Your lab scores all count, but your lowest lecture score. So if you really struggled with one of the lecture exams, something happened, something came up, you have that opportunity to show me that you gained the information, retained the information, and I will reward you for that by replacing your lowest lecture score with the final exam score. All righty. Questions on any of that? And somewhere along that way, I'm guessing we will squeeze in at least 1 more daily quiz. I'm guessing we'll have at least 1 more of those. So again, always important to be here on time. All righty. Any other questions on our game plan for the next few weeks? All righty, excellent. Blank screens, silence. You know how much I love those things. Excellent. All right. As I mentioned, we are going to start by working our way through the rest of the brain anatomy that we were talking about. Excuse me. We had worked our way through the telencephalon, through the diencephalon, and through the mesencephalon when we left off. Excuse me. So we are now on to, whoops, not that button, perfect. The metencephalon. The metencephalon is inferior to the midbrain, and there are two big, massive enlargements to this region. One of these is the anterior enlargement that we see here. This is a structure known as the pons. And the second is the posterior structure known as the cerebellum. That cerebellum is that little nugget on the back of the brain. Let's talk about the pons first. The pons, as this illustration shows pretty nicely, is comprised up, like most of the brainstem. All of these structures here are the brainstem. Basically, as we've seen already, have two components to it. They have white matter pathways, and those white matter pathways are basically carrying information from one location to another. And then they also have nuclei, which of course are the clusters of gray matter, or more specifically, they are the cell bodies and dendrites. Of some type of cell bodies and dendrites of the axons, of the neurons, I should say, clustered together for some function. Here in the pons, for instance, we see a pneumotestic center and an apnotestic center. These play an important role in respiration. And some of the other basic body functions occur in nuclei located in this area. But as you can also see from this illustration, there are two pathways of axons. There are axons that are traveling to and from the cerebrum, to and from the big telencephalon that we have on top. But notice also over here to the side are these pathways that go from the brain to the cerebellum. And in fact, if you look closely here at our illustration, you will see is there are not one, not two, but there are indeed three. Actually, I probably should have done that in a different color. Yep. Big cut bundles of axons that are going to our cerebellum. Of these, one is the largest of these. There is the one on top, which of course, as we know, is going to be the superior. The one that's considered on the bottom, which is going to be the inferior. And the one in the middle, which is known as the middle. This middle bundle of axons carries information to and from the cerebellum. Kind of like this fiber tract up here carried information to and from the cerebrum. The one that's went to and from the cerebrum, we called the cerebral peduncle. Well, guess what we call this middle structure, bundle of axons going to the cerebellum?
The middle peduncle?
True, middle cerebellar peduncle, because we want to make sure that we know it's going to the cerebellum. Absolutely. And you could have either A, deduced that from the huge clues that I give you, or B, from looking at the list of anatomy terms that you have right in front of you, because that's the only one you're responsible for. So notice as we look at these, and I think I have one more picture. Yeah, here we go. Excellent. Notice this, as it says here on the illustration, is a posterior view. That doesn't show up. Is a posterior view. And so with that posterior view, you get a better sense that one of these is on top, one of these is on the bottom, and then the big one right here in the middle cut going to the cerebellum, which would basically be sitting in front of us, is the middle cerebellar peduncle. And again, just a bundle of axons carrying information from the brain to our cerebellum. Let's talk about that cerebellum. And to do that, let's start first with this. Here is a very simplistic illustration of our brain and the beginning of our spinal cord. As we have already talked about, we know this big, huge mitten-shaped structure is the telencephalon, known as the cerebellum, that has that gray matter and white matter that we talked about, the cortex and the limbic system and all those things that we discussed. Beneath that, we know the diencephalon and the mesencephalon are buried deep inside of this and we don't see it. But here we see the two structures of the metencephalon. The enlargement that is the pons here to the front, and then here on the posterior and inferior part of the brain is a structure known as the cerebellum. That cerebellum, and I'm going to cheat, and I think I'm going to bounce back and forth between the two slides. Here we go. We see it in a cross section here. Here I like this view nicely. When we look at the gross anatomy of it, we see that like our cerebrum, the cerebellum has two hemispheres, hemisphere 1 and hemisphere 2 over here like that. But you will notice, unlike the cerebrum, which has this huge, deep invagination, and can somebody remind me what that big, huge invagination that separates the two hemispheres of the cerebrum is called again?
A central sulcus?
No, not the central sulcus, no.
How about occipital sulcus?
Nope. Come on, peeps. A, you got a list of anatomy in front of you. B, we've been going over this a lot. What is this big, huge? There we go. The longitudinal fissure. Absolutely. Excellent. That big, huge indentation that separated them. However, if you notice here with our cerebellum, and I know when you look at it over here in an actual cerebellum, it kind of looks like a pound of ground beef the way it looks there. But if you look closely, you will see right here on the midline is a raised bumped region. Notice here the illustration shows a little bit nicer. And again, because of all the nooks and crannies in the cerebellum, it has a segmented appearance. So we have this long, thin, segmented looking structure that almost looks a little bit like a worm down the center, a raised ridge instead of an indentation. And any idea what we might call that raised ridge on the midline between the two hemispheres of the cerebellum?
The vermis.
Vermis, excellent. And how did you know that? Did you know that because vermis comes from the Italian word for worm and because it has a large segmented looking structure?
Yes.
Excellent, that's what I thought. However, for less clever individuals, you might have gotten it from right here or from your anatomy list that is sitting in front of you as well. Now, remember also up here on our cerebellum, we have raised ridges and we have grooves. And what did we call the raised ridge again here on the cerebellum?
Is that the folia?
No, for the cerebellum. For the cerebrum, I'm sorry, for the cerebrum. You're right, for the cerebellum it is, but for the cerebrum, what would you call it in the cerebrum?
The gyrus.
Gyrus, absolutely. It is a gyrus. And then the indentation, what did we call the indentation? Oh, don't feel bad, Yulia. I'm going to make up for it by probably having your next daily quiz on Thursday. And since it's the last one, I'll probably make it 10 points just to make sure you guys are prepared for that. So don't feel bad. Excellent. Sulcus. Perfect. We have that raised ridge, which is a gyrus. We have that indentation, that is the sulcus. Well, notice we have the same kind of anatomy here on the cerebellum. And sure enough, the indentations between, it's not going to work. Cheat and do it this way. The grooves that are located here are indeed still called a sulcus. But you're right. In this case, the raised ridges are not called gyri. Instead, it is called a folium. Plural being folia, because they look like the ruffled edges of a leaf. So they are called folia. So both structures have sulci, but here on the cerebellum, they are folia. Now, of course, one of the challenges for this then is how are we going to distinguish them under the microscope? Because you absolutely positively are responsible for this stuff under the microscope. And I know we haven't had too much of a chance to talk microscopy. Hopefully you've looked at that histology lecture that I've posted for you to have the opportunity. If we have time at the end of classes, I will do some more histology for us, but if not, make sure you look at it because you're absolutely positively responsible for this stuff. But let's go ahead and take a look at the cerebellum here histologically. Here we have a low magnification view, and sure enough, lo and behold, we see some raised ridges and we see some grooves. And so as we know, these raised ridges are known as a folia, or folium, let's do the singular here. And our groove is indeed a sulcus. So then the question becomes, how can we tell the difference between the cerebrum that we were looking at before and the cerebellum? And it's by looking at the difference between the gray matter and the white matter. In both the cerebrum and the cerebellum, and here, let's go ahead just to avoid confusion so that I'm not labeling anything. Let's put a box up here so that we can write in it. In both the cerebrum and the cerebellum, white matter is deep and gray matter is superficial. Okay. Oops. White matter is deep and gray matter is superficial. All right. And that's what we see here. But what is special about the cerebellum is that it has two layers of gray matter. So if you look closely here, what you will see is this light colored material is one layer of the gray matter. This dark layered material is the second layer of the gray matter. And then in between them, deep to them, is our white matter. So this structure that I'm drawing in blue is our white matter. Now, the two layers of gray matter are known as the cellular layer and the molecular layer, and you don't have to worry about distinguishing which is which. But if you notice, this white matter underneath it forms a big, elaborate, one might almost say tree-like looking structure. So collectively, all of this white matter of our cerebellum, what do you think we call it collectively? Now, the white matter we don't call gray matter. This white matter forms a big, elaborate, almost tree-like looking appearance. Let's go back to our other slide.
Is it the cerebral white matter?
Nope, cerebellar white matter. Notice here in our cerebellar white matter, the white matter is deep. It forms this big, elaborate, branching, almost tree-like looking appearance. There you go. Madison's got her list in front of her or has read ahead. It is the arbor vitae, which means the tree of life, right? Because the cerebellum plays a very important role in many of the functions of our body. It has a tree-like appearance. It's a tree in your brain, so it's the arbor vitae, the tree of life. Excellent. So we have this big elaborate tree of life, this arbor vitae, which forms the white matter. When we look at the gray matter, as I mentioned, there are the two layers of it. And while you do not have to distinguish the two layers by name, you don't need to know which one is the cellular and which one is the molecular. The reason I keep emphasizing this is because as you also look on your list, you will see that there is a particular unique cell that is found here in the cerebellum. And that cell, if you remember, we talked about once before, was the Purkinje cell. Right? That Purkinje cell is located here. Here's another beautiful, beautiful view. Again, notice different stains. This stuff always looks differently, but notice we have our white matter here at the center. We have two layers of gray matter 1 and gray matter 2. And what's nice about this is here at the border between the two layers of gray matter, what color do I want to use for this? Let's use brown. You see all these cell bodies. All of these cell bodies you find at the boundary of the two layers of gray matter are our Purkinje cells. So notice here we have a location. The cerebellum in between the two layers of the gray matter. We have a location and a name for the specific cell, a Purkinje cell. Now, based on its location in the gray matter of the cerebellum, what do you think its function is?
Wouldn't it just be an interneuron that communicates between the other two neurons between motor and sensory?
Absolutely. It is and it doesn't even have to be between motor and sensory, but you're absolutely right. It is in between. So cerebellum is between the layers of gray matter. So you are right. From a functional standpoint, that makes it an interneuron. And if we know it's an interneuron, what must its structural classification be?
Multipolar.
There you go. So notice how one piece of information, the location, one piece of information, the location, gave us the functional classification and the structural classification. Function, structure, location go hand in hand in hand. The reason that, okay, great question. The reason that the gray matter and white matter are different colors is because different stains are used. They're simply just using different stains that emphasize different things. This particular stain happens to stain more of the myelin. So not only are you seeing the cells, but you're seeing more of the axons and stuff that are coming out of those cells to form the gray matter, whereas the previous one didn't. Remember, gray matter, when we talk about gray matter and white matter, we're really talking about the anatomy of it, the parts that are made up of cell bodies and dendrites, or the parts of it that are made up of axons. When we're looking at color, that's something that is added artificially to it. And so different stains are going to look different ways. Yes, there's another question. Yulia.
Yeah, didn't you just say that both the stuff that stained blue is white gray matter and the stuff that stained white is gray matter?
Yes, but notice I've colored it, so let's erase it. Notice if you look here, the darkest thing is actually the white matter. So
okay, I'm just making sure I'm understanding you correctly.
Yeah. So don't worry so much about what the color is, it's the relationships.
Okay.
In the cerebellum, we basically have 3 layers of stuff. layer 1, layer 2, and layer 3. The deepest thing in the cerebellum is always going to be the white matter, no matter the color. And then layers 1 and 2 are going to be the gray matter. And in between them is where you're going to find the cerebellum. Notice we go back to the previous picture. This is a much, much lower magnification, but we can clearly see the deep layer, which is the white matter. We can clearly see the 2 layers of gray matter. And even though we can't see it at this magnification, we know there must be cell bodies located right here at the border of these 2. And that would be the Purkinje cells. We're just not seeing them at this magnification. Yeah, it's the tiny convoluted region underneath the cerebrum. Absolutely. So the cerebellum is under the cerebrum, posterior and inferior of the cerebrum. Now be careful, they both make up the brain. The cerebrum is not just your brain, the cerebellum is part of your brain as well. So yes, if we go back to this picture, this whole thing that we're seeing here is the brain. The largest part of the brain is the cerebrum, right? That's this big, huge mitten-shaped thing formed from our telencephalon. The next biggest thing is the cerebellum. So these are both parts of the brain, but different locations with different structures and different functions. I think I have one more picture. So I have that one, I have that one. Oh, this one's even more. So notice this one, we are just looking at the gray matter. Obviously, without the white matter, it's a little trickier to see what we're looking at, but we can clearly see the 2 different styles of gray matter, and we see these big, huge cells that are indeed multipolar. Now, I know when we look at them here, they don't necessarily look multipolar, but remember, there was something special about those Purkinje cells. Obviously, they have one axon that comes out of it. So when we cut them, what are the chances we're going to see that one axon, right? It's like if you throw an apple up in the air and you close your eyes and slice it with a knife. What are the chances you're going to cut perfectly through the apple where the stem is still attached to it and you can see the stem coming out? It's not very likely to happen. And that's what the things are going to be like with this axon. But what you can see some of is where those dendrites are coming off. And if you remember when we talked about it, the dendrites of these Purkinje cells are these big, elaborate, fan-like dendritic trees. We don't particularly see that with this particular stain and this particular magnification, but there's this beautiful picture of a colorized. Oh, great question. No, actually, that's a blood vessel. That's actually that thing you see up there at the top. That's actually a blood vessel. So we're not seeing, there's no white matter that you're seeing in that. That's actually a blood vessel. Here is a colorized electron microscopy view. So using an electron microscope, what they're able to do is actually inject these cells with fluorescent dyes. And when you inject them with these fluorescent dyes, then you're able to see how that cell has this big, huge, elaborate dendritic tree, or this one's got an even nicer one that you can see coming out, forming this big fan-like structure here in the outer layer of the gray matter. And then, of course, this here in the center is our white matter, the axon tracts, and then the 2 different layers of our gray matter. And all these cells lining up in the middle are those Purkinje cells. So we have a location between the layers of gray matter in the cerebellum. We have a function, interneuron. We have a structure, multipolar. All right, so the 3 go hand in hand in hand. All right. Excellent. So we have identified the cerebellum. We have talked about the anatomy of the cerebellum. We have talked about the microscopic anatomy of the cerebellum. What we haven't talked about is the function of the cerebellum. The function of the cerebellum actually has many functions, but one of the primary ones, and the ones that we're going to focus on, is that it plays a very important role in 2 things, motor stuff. The first is maintaining our balance and posture. I know that seems like a really, really basic thing, right? All of you right now, hopefully, could stand up with your eyes closed, look up to the sky, and be able to maintain your balance. unless you've had a little too much Jägermeister for breakfast. If you've had too much Jägermeister for breakfast, is it as easy to stand with your eyes closed and maintain your balance? No, we don't think
No.
about how complicated that task is until it's impaired. And that's kind of what it's responsible for. It maintains that balance, it maintains our posture, and it helps to maintain learned behavior. Like walking. Again, your cerebellum's not responsible for initiating walking. It can't terminate walking. It can't get you started. It can't make you stop. But what it helps you to do, kind of motion memory is part of it, but it's actually helping to refine that learned behavior. Remember, one of the things that we've talked about is one of the ways we can easily see how these systems work is to see what happens when it's impaired. When your cerebellum is impaired, it's a condition we call ataxia. And it's similar to what it appears to look like when you are drunk. And this is actually what has happened many, many times. What happens is someone is in a car accident. The police show up, they ask the person to get out of the car, and the person stumbles getting out of the car. The person's wavering back and forth while they're trying to stand there. The policeman asks them to walk a line, and they're stumbling all over the place. So they arrest them on suspicion of drunk driving. They take them back to the precinct, and they have them blow a breathalyzer test, and their blood alcohol level is 0.00. Well, what the heck's going on? Well, it turns out what often happens, actually more frequently than you would think, is when that person was in the car accident, they had the headrest that wasn't properly aligned to their head. And when their head got whacked back into the headrest, they had a major trauma to their cerebellum. And that major trauma disrupted that ability to smooth out the learned behaviors, right? To refine those learned motor patterns. And so as a result of it, they were stumbling all over the place, wavering all over the place, couldn't walk a straight line, couldn't stand up straight, not because they were drunk, but because they'd had a traumatic brain injury. They had an ataxia caused by impairment of function from damage to the cerebellum. Now, in most of the cases when that occurs, it's a temporary trauma, and most people recover from it relatively quickly. However, in very rare instances, it can be permanent. Imagine what that would be like. All of the problems of not being able to maintain your balance, not being able to walk a straight line, not being able to stand up straight without any of the fun associated with the alcohol. You'd basically be in a constant state of being drunk without actually being drunk, where you'd lose that ability to refine those motor skills, and you'd have trouble walking and standing, playing the piano, or any other type of learned behavior. Again, it doesn't help you when you're first learning to play the piano. But once you've learned to play the piano, you're able to play the piano and have a conversation and do things along those lines and be able to get it. It wouldn't be so much vertigo, although because vertigo has more to do with the balance areas of your sensory systems that we'll talk about. So you wouldn't get vertigo, but it might be possible where you might get some motion sickness, because if you don't necessarily know the direction your body's going to be moving, that disruption in the smoothness of that may potentially, the same way like being on a boat could make you seasick. So there might be some seasickness that would go along with that. I'm not certain about that. I'm totally guessing, but it wouldn't be vertigo in the same way. That has to do with more equilibrium and balance. Yes, there was another question. Yuli?
Yeah, I have a quick question. Did you say general damage or injury to the cerebellum or a specific region?
No, just general to the cerebellum.
Okay, and then my second question is Pons and cerebellum are two different sections of the.
Yes.
Okay.
The way I remember that this is the metencephalon is, met has the T in it. And so notice the T, the metencephalon has these two enlargements. The one enlargement to the front, that is the pons, and the one enlargement to the back, which is the cerebellum. And notice one other nice thing about this illustration. We haven't fully talked about them yet, but we did mention how there are fluid-filled spaces in the brain. Notice between the pons and the cerebellum is our fourth ventricle. So there is actually a fluid-filled space here between the pons. So it's the pons to the front, cerebellum to the back. So these are two completely different neural structures and a fluid-filled space in between them. All right, excellent. I think that is everything on our metencephalon. Any questions on that? Anything I missed?
Nope.
All right, excellent. So that brings us to the last our last cephalon, our last enlargement of the brainstem, and that is the myencephalon. The same way the telencephalon is the cerebrum, the myencephalon is the medulla oblongata. So this inferior part of the brainstem is known as the medulla oblongata, and it is formed by the myencephalon. Notice again, as we see here from the illustration, like we've talked about, all these brainstem structures have a large number of axon tracts and a large number of nuclei. Notice also, as we've been working from the top of our brain all the way down to the brainstem, the function has gotten much more basic, right? So up here, we have areas of the brain that we talked about that respond to just a particular movement or the particular identification of a visual item or color or smell or things along those lines. Whereas down here in the medulla, we have the most basic functions. Here in the medulla is where we have nuclei that play a role in controlling our heart rate, in controlling our respiration rate, basic living functions that us and every other living organism does, like coughing, vomiting, swallowing, hiccuping, sneezing. All of these basic temperature regulation, all of these things are controlled by nuclei located here in the medulla oblongata. This is the base of our brainstem and the most basic functions. Now, I want you to notice one other big important thing we're going to be talking about a little bit later today. Here in the medulla oblongata is also where we find over half of our cranial nerves. Again, you don't have to know this yet, but this is 6, that's 7, this is 8, that's 9, this is 10, this is 11, and this is 12. And we have the same thing on the other side as well. So notice the majority of our cranial nerves also are associated with our medulla. I point these out to you now, not because you need to know the names of them or identify them or know anything else about them yet. But if you look closely at how all these cranial nerves are aligned, what you will notice is that associated with them are 2 big enlargements. This first big enlargement that are paired, one on each side, are what are known as the pyramids. These pyramids are the enlargement where the majority the pyramids are where a majority of the axons, say it that way, cross contralaterally. Remember, one of the things that we've talked about a lot is if I want to move my right hand, I make that decision here on the left side of my brain. If I want to feel something with my right hand, I process that information over here on the left side of my brain. So both sensory and motor information crosses contralaterally. And it is here in the pyramids where most of these axons cross, where things coming up the spinal cord from the left go to the right, and things that are coming up the spinal cord from the right go to the left. Here in the pyramids is where most of our axons cross. Again, I said most, not all, but most of them cross. Something like 90% of them cross here in the pyramids. Now, the other thing that I want you to see here, and we'll take some other looks at this a little bit later. The second big enlargement, this big bump that sticks out right here between cranial nerve 12 and cranial nerve 9, 10, and 11 is this big green circle that they're representing here as well. It is what is known as the olive. And the olive is what gives us our sense of proprioception. We talked about this proprioception when we talked about combining information about proprioception with our vision and our auditory information. Does anybody remember what proprioception is?
So awareness of where you're at in space.
Yeah, it's the awareness of where your body is in space. Again, this is another thing we take very much for granted. Because again, unless you've had too much Goldschlager for breakfast, you can close your eyes and you can touch your nose with your fingers. When you wake up in the morning, even before you open your eyes, you know how your body is oriented, laying down in the bed, head towards the foot of the bed, where the bathroom is in relation to you, all of those kinds of things, right? Until you wake up from that Saturday night at some random house, and then you get this whole big warping of your sense of where you are in space because of that. And again, it's a thing we take for granted, kind of like we take our voice for granted. As we've talked about before, the ability to speak is by voluntarily controlling skeletal muscle in our throat and neck, right? It's not, I am voluntarily controlling it, but I'm not actually consciously aware of what muscles I'm putting tension in. However, as we talked about, when that boy enters puberty and his vocal cords thicken, the muscles that he normally would contract to put tension in it to produce his speech don't produce the same types of vibrations that they did before because of that disruption in the size. That change in the size changes the vibration, and he has to relearn how to put tension in the vocal cords. And what happens to his voice while he's relearning that? Exactly. His voice cracks as a result of it. Well, it's the same thing. You've got this teenage boy walking down the hallway, and there's that vase that has been here in your family for 14 generations. And he's just walking down the hallway, and he whacks it with his arm and knocks it to the floor and breaks it. Right? Or maybe he's just walking down the hallway and he falls. Right? Did he knock the vase off because he's an asshole? Did he fall because he's on drugs? Well, maybe he's an asshole, maybe he's on drugs, or maybe it's the fact that he's grown 6 inches in the past 3 months. And as a result of that, he's got less awareness of where his body is in space. And so while he's relearning to coordinate and understand where his body is in space, he may be a little bit more awkward. He may be a little bit more clumsy as a result of that. Again, this can happen to females as well. Boys tend to have more dramatic growth spurts. So you see it a little bit more commonly, that disruption in boys. The same way, again, the voice cracking is something that primarily just happens in males. That is because testosterone affects the vocal folds more strongly than the estrogens do. So that is something that is unique, but males tend to have larger growth spurts, but a female that has large growth spurt can have those same types of coordination problems where they're a little more clumsy, they're a little bit more less aware of where their body's in space, so more likely to knock something over or to trip and fall or something along those lines as well. There's so many of these complex processes that we take for granted when they work properly. Right? It's when they're disrupted that we're really aware of how important that they are. All right. Yes.
Yeah. Could you remind me what the function of the pyramids is?
Again, it's a bundle of axons. So axons are really just pathways. So they don't have functions per se. What's special in this case about the pyramids is this is where the majority of our axons cross from one hemisphere to the other, or from one side of the body to the other. Because as we know, most of our control, most of our sensory information is contralateral.
Mhm.
We process hearing from the right ear on the left side. We move my right hand on the left side. We touch with my right hand on the left side. All of those things. So at some point, axons have to go from the right side of my body to the left side of my body to get to the left side of my brain. And here in the pyramids is where most of that occurs. All right. Excellent. With that, we are done with the myencephalon. And that is the brain anatomy I want to cover for now. How are we doing on time? We're doing okay on time. Excellent. So those are our 5 cephalons. Go ahead and stop that. Where do I need to go? Give me a second to figure out what I want to do next. I'll save that for later. I think I'm good with that. All right. So, as I mentioned, here we have that original picture that we looked at. This nice illustration from your textbook that shows our 5 cephalons. We have our telencephalon, we have our diencephalon, our mesencephalon, our metencephalon, and our myencephalon. We have identified all 5 of those regions, identified the structures that form them, and in each of them we've identified some type of area or region, gray matter nucleus or the cortex that again is that functional component that has some specific function. We've identified them and what they do. All right. What we have alluded to and haven't talked about fully and need to do now is how we protect all of this stuff. I'm sure you remember because it was on the very first day of class. And on that very first day of class, I'm sure everything I said has been completely crystallized in stone in your brains. But if you remember way, way back in the day, we talked about how the structures in the dorsal body cavity, the cranial cavity and the spinal cavity, those were protected by a structure known as the meninges. Right? Remember, this is different from the ventral body cavity where everything is protected by a serosa. Well, as we hinted at that point, the meninges are actually made up of 3 distinct layers. And each individual layer, meninges is actually the plural, the singular is meninx. The first meninx, yes, I did tell you to do that, and again, shows how wise you guys are because you didn't. So, or stubborn, or dedicated, but you know that could be a good thing too. Absolutely, excellent. I never lied to you guys. I've always was honest with you guys about what your expectations are. Right. So again, let's talk about the 3 meninges that make up our protections, outer protection of both our brain and spinal cord. So the first is a double layer of very thick, dense, irregular connective tissue. This double layer is a big, huge, thick, tough mother, which is exactly where it gets its name. It is known as the Dura Mater. Now, I want you to notice something closely about this Dura Mater. Notice if you look closely, this first layer of dense irregular connective tissue is actually connected to and surrounding the bone. what would we call a layer of dense irregular connective tissue that was on the outer surface of the bone?
The periosteum.
Exactly. So notice the outer layer of the Dura Mater is the periosteum. Notice what this does is this directly connects this protective bag that is going to surround and protect the brain to the skull. Now, why would you want to connect this to the skull? Don't our bones move around?
Not these ones.
Not the
Not these
skull.
ones, absolutely. Not the skull bones, absolutely.
Fissures.
So these are immovable bones forming a solid, immovable structure. And so it makes sense to connect our protection to it. And so indeed, that outer layer is actually the periosteum. But notice there is also an inner layer as well. This inner second layer. But I want you to notice something else interesting about these two layers. Notice over here, they are connected to each other. But if you look closely, right here where we get to the midline of the brain, and notice here, this is our longitudinal fissure again. That separates our brain into two hemispheres. Notice here where the longitudinal fissure is, these two layers separate from each other. And when they separate from each other, they form a fluid-filled space. And that fluid-filled space is actually more than that. That fluid-filled space is a big, loose vein. And we have a big fancy term for a big loose vein. Anybody know the term we use in the body for a big, really loose vein? It's called a sinus. What's the advantage of having a really big loose vein?
Things can move through it easily.
Absolutely, right? Again, we have a tremendous amount of blood going to all parts of our body and coming away from it. And one of the things we'll learn when we get to our cardiovascular system is that our cardiovascular system in 431 is leaky. So a little bit of water comes out. Every time blood goes to a capillary, for instance, in your hand, a little bit of water leaks out. But luckily, we have lymphatic capillaries, our lymphatic system, which collects that and helps to drain it back and put it back into our heart. And if it doesn't do a good job, or if I get an increase in blood to my hand because maybe I smash it in the car door, what happens when either I get an increase in blood flow or I have problems draining it?
Inflammation.
Inflammation, swelling. Now, if my hand swells, is that a major problem?
It can be, depends on
Depends on
much,
how
but not a
severe it is.
huge deal.
Yeah, it's got to get really huge, right? I mean, normally when my hand swells, the biggest problem I have is getting that wedding ring off when I get to the bar on Friday night, right? It becomes a little harder if your hand's swollen, right? It may be a little tight as you're trying to, but it's not a major issue. But what about here inside of this non-moving skull? Do we want
Big
fluid
issue.
leaking out here and not being collected and not being brought back to the heart?
No.
No,
No.
because swelling here inside of the cranial cavity is a huge, major issue that can cause permanent damage, right? Much, much more severe. So we want to make it as easy as possible to drain all the fluid out of the cranial cavity. So there are large, large venous sinuses that are going to help to collect fluid, both blood and cerebral spinal fluid, which we haven't talked about yet, and help to drain that out of the cranial cavity so we don't get an increase in swelling, an increase in pressure inside of our cranial cavity. So having these big sinuses between the two layers of the dura mater, and so not surprisingly, these are called the dural sinuses. Oops, not dual, dural sinuses, because of the dura mater, are very, very important. Excellent. That is tissue 1. Tissue 2 is this purple looking stuff that we see here. Again, notice this one is a very thin layer, but what's much more distinct about this one is it has these large number of collagen fibers. And because it has a large number of collagen fibers coming out of it, it has a very spider webby appearance. Hold on, let me finish this. So again, cobwebby. That cobwebby appearance is why this layer is known as the arachnoid matter. So, oops, matter. The dural sinus was this fluid-filled space here between the, so let's put it here. Dural sinus is this fluid-filled space between the two layers of the dura. That's what the dural sinus is. So this here is a big loose vein for collecting fluid between the two layers of the dura mater. So that's why it's the dural sinus, because it's found here between the two layers of the dura mater. Yuli, you had a question as well?
Yeah, you pointed out the second layer that was right after the periosteum, or right after Dura Mater, but you never gave us a name. Are you still getting to that, or did I miss it?
Oh, it doesn't have a name. So again, let's do it this way. No, not pink. The Dura Mater is comprised of 2 layers of dense, irregular connective tissue. One of those happens to be the periosteum of the bone. So this is the outer layer of the Dura Mater, but it's also the periosteum. This is just the inner layer of the Dura Mater. It doesn't have another name. So there's an outer layer of Dura Mater, inner layer of Dura Mater, but here in the skull, this outer layer of the Dura Mater is also the periosteum. We'll see in the spinal cord that is not the case. Well, you're right. Here it is. Often these things are given 4 different names for one structure, and it can be confusing this way. However, as we'll see, the spinal cord has similar protections, but it needs a different arrangement, because while we just finished talking about the bones of our skull don't move, what about the bones of our vertebral column? Do they move?
Yes.
Yes.
Yeah.
So we need here, we can just worry about protections in a structure that doesn't move. When we get to the spinal cord, we have to protect the spinal cord in a structure that changes shape. So while we're going to still have the same 3 layers of protective tissues, those 3 layers are going to be arranged very, very different. Well, not very differently, but there's going to be some significant differences between what we see here in the skull and what we see in the spinal cord, because here, no movement, spinal cord movement. Okay? So here that outer layer is both the periosteum and the outer layer.
How far does that inner layer go into the longitudinal fissure? Like all the way through
Pretty
it?
much all the way down. In fact, we'll actually see that this is not the only place that the inner layer folds in. There are actually going to be 3 main invaginations of that inner layer, and we're going to give names to all 3 of them. So I have a different picture to show that to you. So yeah, great question, but I will show you that. We'll get there. All right, so again, our second layer, and I think I have to move this, so I'm going to cheat, and let's put it over here. Second layer, cobwebby layer, and again, that cobwebby layer we call the arachnoid matter because of its cobwebby appearance. And notice, because of that cobwebby appearance, it basically produces a fluid-filled space forms a space, and this space is known as the subarachnoid space because it's below the arachnoid matter. And the subarachnoid space contains our friend, the cerebrospinal fluid. Another one of the important protections of our nervous tissue. This fluid that is filtered out of our blood to fill this space and cushion the blood, cushion the brain. Provide oxygen, provide nutrients, provide chemical signals, help to remove wastes, but also form a fluid cushion. Anybody remember the neural glial cell that is responsible for helping to produce the cerebral spinal fluid? There are only 6 you had to learn. Which one of the 6 you had to learn was the one that was responsible for helping to make the cerebral spinal fluid? Anybody remember? Were able to look back at their notes and figure it out?
Yeah, the ependymal cells.
Ependymal cells, excellent. Perfect. All right. Lastly, if you look closely, what color should I use for this? I'm going to go ahead and use black for this. Notice if you look closely, the third layer is this layer right on the surface of the cerebrum. Notice, like the cerebellum, our gray matter is superficial, our white matter is deep. We see that. And notice here, the sulci are kind of fused together. The blood vessels are held in place. And there's basically this shrink wrap that wraps around and stabilizes the nervous tissue and holds it in place. Remember, as I mentioned, this nervous tissue inside of your brain has about as much structure as Jello. So it doesn't have a lot of integrity. So having this shrink wrap wrapping around it helps to stabilize it, helps to hold it in place, helps to keep the blood vessels in place. And this shrink wrap layer on the outer surface, we call the pia mater. So 3 layers of protective coatings, 3 individual meninges that work together to provide these 3 layers of protection. And really 4 if you think about that cerebral spinal fluid in the subarachnoid space. So all of these things bathe the outer surface of our brain, cushioning it and protecting it inside of the skull. All righty. Questions on that?
Is there anything notable that we should know about the PM matter?
Just that it is in direct contact with the cerebrum, stabilizes it, and and stabilizes the blood vessels, holding it in place. I think that that's sufficient.
Wait, did you already go through 69 and 71? No, the falx cerebri.
No, we're getting there. We're getting there.
Oh, okay. I thought I skipped it back.
We're getting there. No, it's a great. I appreciate you checking because it is possible that I missed things on the list, but I wanted to start with that nice illustration. And then notice we can look at a couple things here. Notice here we see an actual brain, an actual skull. Notice here we can see where the skull is and where the scalp and where the hair is. And notice this really thick, tough, fibrous looking leathery material is the Dura Mater. If you have to have brain surgery, they have to cut through it. It does have a consistency like leather. So they can get to the brain, they can do the surgery, and then they can sew it back up again and it will hold. It's a big, huge, thick, leathery structure. Dura Mater, it means tough mother, right? Notice also here we see those sinuses we were talking about, those dural sinuses where the 2 layers separate and there's some blood vessels there in between. Notice they've cut and removed the Dura Mater over here on the left-hand side. Arachnoid matter is very, very flimsy. Doesn't have a huge consistency to it. You can see, if you notice your illustration here from your textbook, Down here, if you look closely on the brainstem, you can see this little fuzziness to the side. I'll draw it here and draw it here and then remove that so you can see it. That little fuzziness that you see around that tissue, that's the arachnoid matter. But notice if you look at the brain itself, the cerebrum itself, we can see some blood vessels that are held in place, and it has this nice shiny surface. The reason it has that nice shiny surface is because it still has that PM matter intact, stabilized and in place. In fact, here, let's do this. Stop that for a second. Oh, I closed it, didn't I? Shucks. There. If you think back to when we first started talking about the brain anatomy, oops, hold on. Do that. Way back in the day, I showed you this picture when we were talking about the lobes, frontal lobe, occipital lobe, parietal, temporal, all of that. Notice this one has that shiny surface, has those blood vessels stabilized in place. This one has that shrink-wrapped pia matter on top of it. However, remember, I also showed you this picture. Notice here on this picture, the pia matter has been removed. Notice there's still some deep blood vessels we can see here, but notice now you can see all of the sulci are kind of open. All of the gyri are kind of separating from each other because they don't have that shrink wrap holding it tightly in place. So here, and notice the nervous tissue is not damaged. They have basically just peeled that pia matter, peeled that shrink wrap off. So here we see it without the shrink wrap, without the pia matter. Here we see it with the pia matter, with the shrink wrap. No, pia matter does not help in the cerebral spinal fluid. We'll talk about what makes the cerebral spinal fluid in just a minute, but where the cerebral spinal fluid is found, let me go ahead and close this because I have too many things open. Where the pia matter is found is in the subarachnoid space. It's not made in the subarachnoid space, but in the subarachnoid space is where we'll find it. We'll talk about where we'll make it in just a minute. So in fact, the cerebral spinal fluid would basically be between the arachnoid matter and the pia matter in the subarachnoid space. All right, excellent. Here is another great view of a cross section through this and a nice illustration. And again, there's a couple key things we can see here. Notice again, we have the 2 layers of the Dura Mater. This first layer is the one that is part of the periosteum, the surrounding the bone. And then this one here. Notice next here in green, we have the arachnoid matter with its collagen fibers, and then ultimately connecting to the pia matter, which is the shrink wrap on the gray matter. Now, as we've talked about, this here is an actual space, and this actual space contains cerebrospinal fluid. I make a point of emphasizing it because we have another space here. Notice here, between the Dura Mater and the arachnoid matter is a potential space. This potential space is called subdural space. Now, what do I mean when I use the term potential space?
Sometimes it's there and sometimes it's not, or it gets filled with something.
Okay, I like that. That works. Absolutely, right? I like that. That's a pretty good definition. How many hands do I have?
2.
2. When I mash them together like this, how many hands do I have?
2.
Two,
Still have 2
but
hands,
acting
absolutely.
as one.
Well, is there, ask the question this way. Is there anything between them?
No.
No, there's nothing between them. But just because there's nothing between them doesn't mean they've fused together to become a single thing. Basically, in between them is what we would call a potential space. You are right. That potential space could fill with something, but in normal healthy individuals, your Dura Mater and your arachnoid matters are smashed up against each other, so there's nothing in between them, and they're just a potential space. And in a normal healthy individual, that would be the case. If, however, you had some kind of traumatic brain injury, then what could happen is some blood could start to accumulate in this space between the arachnoid matter and the Dura Mater. And what would we call that enlargement of blood in that space? I know you know.
Hematoma?
What kind of hematoma?
A cerebral hematoma?
Close. It's in the subdural space. So what do you think we call a hematoma in the subdural space?
A subdural hematoma.
There you go, a subdural hematoma. Absolutely. So in a normal healthy individual, there should be nothing in this subdural space. It should be just a potential space. But with a traumatic brain injury that involves bleeding, it is potential where there could be a subdural hematoma, right? Even if you've never known anybody who's had one, all you've had to do is watch some kind of medical drama or general hospital or anything, medical related. And I'm sure at one time or another on that show, they've wheeled somebody into the emergency room with a subdural hematoma. Right? Well, that's what's happening. They have blood that's not supposed to be there accumulating in this space between the dura and the arachnoid matter. All right. Excellent. Questions on that? All right. Last thing I want to do before the break to answer that question from a while ago. As I said, there are 3 big invaginations of that inner layer. So again, as we talked about 3 times, or in 3 different locations, let's say that. The inner layer of the dura expands out to stabilize the brain. And when it does that, it forms these 3 important structures. The first of these 3 important structures is the falx cerebri. The falx cerebri is the one that we saw that sits in the longitudinal fissure. And it really does go all the way down to the corpus callosum. So it goes all the way down into that longitudinal fissure. The second of these is the falx cerebelli. Notice the falx cerebelli sits between the hemispheres of the cerebellum. But notice it's much more shallow. And the reason this is much more shallow is because if you remember, our cerebellum doesn't have a fissure. Instead, it has the vermis, that raised ridge. And since it has a raised ridge, there's not nearly as much space for it. So the falx cerebelli is much, much smaller than the falx cerebri. Lastly, there is a third layer that sits basically like a tent on top of the cerebellum, and it separates the cerebellum from the cerebrum. And what separates the cerebellum from the cerebrum, like a tent over the top, is the tentorium cerebelli. So these are the 3 Dura Mater enlargements formed by that inner layer of the dura that help to stabilize and hold the brain in place. The falx cerebri, the falx cerebelli, and the tentorium cerebelli. All right. Questions on that?
Just a quick one for clarification. So those 3 that went over are an added layer into the Dura Mater right below it.
So they're an extension. Remember, as we saw, and I'll just cheat, so for instance here, the falx cerebelli, what happens is you have the outer layer of Dura Mater that is in contact with the bone. And then you have the inner layer. And basically what happens with the inner layer is the inner layer extends out. As the inner layer extends out, it basically does 2 things. The first thing it does is it forms that sinus. Notice there's that fluid-filled sinus that we talked about right here, where a space where we can have those big blood vessels to drain the brain and the nervous tissue. And that inner layer extends out to form some kind of structure that forms that protection, that forms that stabilizing structure. Well, notice here, there is no such thing as an epidural space. Epidural space would be above the Dura Mater, but remember the Dura Mater is actually attached to the skull, and because it's attached to the skull, it's not even a potential space. There is no epidural space here in the brain. However, remember, as we talked about in our spinal cord, we need some flexibility of movement, and there you will find an epidural space. But here, protecting the brain, there is no epidural space. It's one of the things that'll be different between the brain and the spinal cord.
Professor, did you say the 3 meninges you just pointed out are an extension of the Dura Mater, or?
Yes, these are extensions of the inner layer of the Dura Mater. The
Okay.
falx cerebri, the tentorium cerebelli, and the falx cerebelli are all extensions of the inner layer of the Dura Mater.
Thanks.
Yep. All righty, excellent. So those are the protective layers. Questions on that? All right, the last thing we need to talk about for protections of the brain are to talk about the fluid, that cerebrospinal fluid, where it's produced, how it circulates, and all the fun things associated with that. But I think this is a good natural stopping point. So let's go ahead and take our first break here, because we got a lot of material to cover. So if we take a good break, then we can stay fresh. It looks like it's 1:20 here, so we will restart at, what did I say? It was 1:20? At 1:35. And at 1:35, we'll restart, and I will start the recording at that time. At that point, we will talk about our last major structures that we need to, or really, these in this case aren't structures. These are going to be spaces, fluid-filled spaces to be specific. I
have a quick question, Professor.
Yes.
Is this when we talk about the movement of the cerebrospinal fluid in the ventricles?
Yep.
Okay, and then I have a second question. I started.
No, you only said you could add. You only said you had one.
I started watching your histology review last night.
Okay.
And you said if we turn in our papers, I'm sure you said it last year, but if we turn in our papers with all of our drawings, you give us 10 extra credit points.
No, I told the class 2 semesters ago when we had been forced online unexpectedly that if they did that they would get the points for that. But no, I'm not providing that here. Here, you have started online. The expectation that is that you've been online. And so, no, unfortunately, that is not something that I'm offering. So,
okay, no. That's it. I'm going on my break.
All right. Sounds good. I will see you guys all in 15 minutes then. All right. See you guys in 15 minutes. All righty. So the last thing we have to talk about for the brain and the brain stem is our last bit of protection, and that involves, again, that cerebrospinal fluid, all one word. Cerebrospinal. Oops, did I spell that right? The cerebrospinal fluid. As we've talked about, its function is, yes, to act as a fluid cushion, a fluid cushion, providing that protection both internally and externally, but it is more than that as well. Cerebrospinal fluid is basically very filtered blood plasma. And so as such, it contains chemical signals that are in the blood, the pH, the temperature, the chemical composition, all of those types of things. Yes, when healthy, it should be clear. There should not be any particulates in it. No, because again, remember the red comes from the formed elements from the red blood cells, and formed elements should not get into the cerebral spinal fluid. So it is just filtered blood plasma. None of the formed elements are in there. As we talked about, it is made with the help of those ependymal cells. Actually, let's put that back. It's made with the help of ependymal cells. So our ependymal cells, which are part of our neuroglial cells, wrap around capillaries, which is where our exchange of materials take place, and we give a fancy word to these capillaries wrapped up in ependymal cells. We call them the choroid plexus. And so it is choroid plexus that makes the cerebral spinal fluid. These choroid plexes are located in these 4 internal spaces we see inside of the brain. Basically, what this illustration is showing us the spaces inside. There are indeed 4 of them. There are the 2 C-shaped lateral ventricles. So there is ventricle 1 here, is ventricle 2. Here, remember where Diencephalon was, is ventricle 3. Notice there is a void here in our 3rd ventricle. That's where that intermediate mass from the 2 thalami connect. And remember, we said our 4th ventricle was located between the pons and the cerebellum. So here we see them from a side view. Here we see them from a frontal view, where again we see the 2 lateral ventricles. And then on the midline, we see the 3rd and the 4th ventricle. No, the 1st and 2nd ventricles are the 2 lateral ventricles. So one isn't the 1st, one isn't the 2nd. They're just the 2 lateral ventricles. They're the ones to the side. So that's 1, 2. And then we have 3 on the midline and 4 on the midline. But you have the right idea. Notice, and you can see it nicely here. Yes, go ahead.
Yeah, are they also the same? I mean, is there a difference between the horn and the ventricle? Are they 2 different structures or the same
No.
thing with different names?
Notice here, so again, that's just a part of it. Notice here, there is this C shape to the lateral ventricles, but notice there's a little bit of a space that projects posteriorly. That posterior bit is considered the horn, but it's still just a part of the 3rd ventricle, of the lateral ventricle. So it's just the lateral ventricle has a C shape with a little horn coming off the back. But it's just that whole C shape with the horn thing is considered the lateral ventricle. So these are spaces that are located inside of the brain. Notice here we see it, although I think we saw it. Here we go. Here's a nice view. Remember here when we were looking at this kind of partial transverse section, we saw the spaces. There's a little bit of the horn that we talked about of that C-shaped lateral ventricle. Remember we talked about how the septum pellucidum separated the 2 lateral ventricles from each other. And then remember how we also talked about how the intermediate mass is in the middle of our 3rd ventricle. So these are things that we've kind of been talking about as we've been working through them. And so inside each of these ventricles, and I'll make this red, inside each of these ventricles is some of that choroid plexus. So it is located here in the bottom part of the lateral ventricles. It is located up here in the top part of the third ventricle. And it is kind of aligned in the center of the fourth ventricle. So if you were a drop of cerebral spinal fluid, you could be made in any of these four locations. All right. So you mentioned earlier before the break how, you know, and it even says it on the handout, the flow of the cerebral spinal fluid. But there isn't truly just one flow, because these could be made in any types of locations. But for argument's sake, let's say we are a drop of cerebral spinal fluid that is produced here in this lateral ventricle. Obviously, our goal is not going to be to fill this ventricle to the point that it bursts like a water balloon that we overfill. We need a way for this fluid to leave. So if you were produced in one of the lateral ventricles, the way you would leave is by this small opening that connects the lateral ventricle. Whoa, that is way too big. That connects this lateral ventricle to the third ventricle. And this space that connects these lateral ventricles to the third ventricle is known as the interventricular foramen. You would then be a drop of cerebral spinal fluid in the third ventricle. Now, if you're in the third ventricle, either because you were made here or because you came through the interventricular foramen from a lateral ventricle, you need to leave, and there's only one way out. You have to descend down into the fourth ventricle. And as you descend into the fourth ventricle, you pass through a space called the aqueduct of the midbrain, or what is also known as the Cerebral Aqueduct. Both are acceptable names. So it's either the Cerebral Aqueduct or the Aqueduct of the Midbrain. That was that hole that we actually saw in the mesencephalon, if you remember, at the center of it. The cerebral spinal fluid. So again, inside each of these ventricles. Oops, why is that not writing? Inside each ventricle is choroid plexus, which makes cerebral spinal fluid. So, following it, we can see all of the spaces associated with the cerebral spinal fluid. Lateral ventricles, interventricular foramen, and that's the only place they can go. If you're cerebral spinal fluid in a lateral ventricle, you have to go out the interventricular foramen into the third ventricle. If you're in the third ventricle, you can't stay there swelling the brain either. You have to leave. So you leave through the cerebral aqueduct, or the aqueduct of the midbrain, to get to the fourth ventricle. And now here in the fourth ventricle, you finally have options. While there is only one way in to the fourth ventricle, and again, that one way in was that cerebral aqueduct, there's not one, not two, not three,